The human brain undergoes rapid growth during the first few years of life. The size of an infant's brain at birth, which is 1/4 the size of an adult brain, rapidly reaches 72% of adult volume within a year. In a developing child, the different parts of the brain that perform a variety of functions develop at different time frames. The part of the brain that governs vision and hearing develops first, followed by parts that govern receptive language and speech. Higher cognitive functions, including memory, executive functions, and emotion, take the longest time to develop. Nutrients play a vital role in regulating brain development during fetal and early postnatal life. The human brain is nearly 60% fat. Omega-3 and omega-6 fatty acids are essential fatty acids that must be derived from the diet. A balanced omega-3 omega-6 ratio is important for optimal health. Since high omega-6 fatty acid consumption in adults has been implicated in weight gain, metabolic and cardiovascular diseases. Docosahexaenoic acid or DHA, the predominant omega-3 fatty acid in the brain, arachidonic acid or ARA, derived from omega-6 polyunsaturated fatty acids, and phospholipids are among the most important nutrients needed for brain development. Fish, marine algae and other seafoods are rich in DHA, which is critical for the growth of nervous tissues. ARA is important for brain health and development. Poultry, eggs and seafood are rich sources of ARA. Phospholipids are a class of lipids that form the major structural component of the brain cell membrane. They are also components of signal molecules, help in neurotransmission, and are involved in maintaining cell membrane integrity. Soybeans, egg yolk, milk, and seafood are rich sources of phospholipids. Myelination is the final process of brain development in which the nerve fibers are wrapped in fatty cells to provide efficient and rapid transfer of information across neural systems. DHA, ARA, and phospholipids help in formation of myelin over the nerve fibers and ensure rapid transmission of nerve impulses. Myelination of the frontal lobe of the brain, the part which is critical for problem solving in children, begins as early as 6 months of age. Thus, the need for nutrients that support myelination is very high during this critical period of brain development. Both DHA and ARA rapidly accumulate in the brain of the developing fetus during the last trimester of pregnancy and in infants until about 2 years of age. Docosahexaenoic acid is also an important membrane component of the photoreceptor cells of the eye, and the optimal functioning of photoreceptor cells is vital for vision. Precursors of DHA and ARA compete for the same metabolic enzymes. In infants, the biosynthetic capability for providing DHA and ARA for brain growth is low, and preformed ARA and DHA are preferentially incorporated into the brain at this time. The newborn is largely dependent on maternal intake of DHA and ARA to meet its nutrient requirements via breast milk. Non-breastfed infants depend on dietary sources for meeting DHA and ARA requirements. The first few years of life are a critical period of brain growth spurt. Optimal nutrition during this period can have a lasting impact on growth and development.